Thank you very much, George, for that introduction, and thank you for inviting me out to Ann Arbor, and thank you all for coming to braving the cold, finally have winter now, and for making it to the first seminar of the semester. Yeasts, of course, are one-celled fungi that don't make fruiting bodies. That's the definition of a yeast. And so yeast are, as all fungi, they are heterotrophs. And of course, heterotrophs are characterized by being lazy and not making their own food sources, and instead taking advantage of all the energy resources that plants have captured from and stored as various carbon compounds. And so carbon metabolism is really the energy superhighway of life. And we can sort of zoom in on the parts of carbon metabolism that are most relevant. We all teach students about the centrality of glycolysis. This is how you take a molecule of glucose and split it into 2 molecules of pyruvate, which you can then run through the TCA cycle and get lots and lots of energy. Of course, there's also the pentose phosphate pathway, which is used to create a redox balance and also provide some protective effects. And we're used to thinking about, especially in multicellular organisms, there being a lot of differentiation based on tissues, and so our livers, for example, take care of a lot of the fatty acid synthesis in addition to detoxifying all of the garbage we consume sometimes. And then our adipocytes actually do a lot of lipid storage. So when things go awry, different things happen, and one of the most famous sort of metabolic problems that organisms encounter is Crabtree-Varberg effect, and this is something that almost all cancer cells actually evolved to do. This is a derived trait, and what happens is they upregulate their hexose transporters that bring glucose into the cells. They upregulate glycolysis and upregulate fermentation enzymes, and instead of making ethanol, they make lactate. And simultaneously, they're downregulating TCA cycle, and so even though they're only getting about 5% of the energy that they would by completely oxidizing it, they're able to make a living just off of fermentation. And indeed, one thing I forgot to mention on the last slide is actually that there are also other specializations in glycolysis. For example, the brain exclusively uses glucose as a carbon source. And we can see in this tumor imaging here, you can see the brain is a real glucose hog. And then the way modern tumor imaging often works is you're actually labeling with a fluoridated glucose, and that allows you to actually see where the tumors are because they're doing glycolysis more. And of course, this is a metabolic defect that cancer cells evolve during our lifespan by misregulating a lot of these same central pathways of glycolysis, fermentation, glucose transport, respiration. And it turns out that Saccharomyces yeasts have evolved to do exactly the same thing over the course of tens of millions of years, within the last 100 million years. Of course, instead of making lactate, I think we can all be thankful that they make ethanol. Nobody would want to drink lactate, although maybe our livers are not as thankful, but it's basically the same process. You still have the glucose transporters are homologous. Of course, all of the metabolic enzymes are homologous, and you still have the same phenomenon where you're recycling NAD and NADH by making ethanol. So the amazing thing, I think, and sort of what George is alluding to, and I don't have a whole lot of results on this project because it's just started, but I think the amazing thing is when we think about our model yeast Saccharomyces cerevisiae, is that this is actually an evolutionarily derived trait, and it's something that most yeasts don't do. Most yeasts do the energetically sensible thing and fully respire glucose all the way to oxygen. And indeed, it's actually within this family of Saccharomycetaceae, even within the family of Saccharomyces, it's actually a subset of yeasts that have actually done this. Almost all of these other yeasts actually prefer to respire. There's at least one exception on here, an independent evolution of aerobic fermentation or aerobic glycolysis in this clade. But this is not a general phenomenon. And so one of the nice things is that, of course, these are all potentially experimental models. They all have relatively small genomes. And we recently wrote a review where we've started to pull together the genome sequence data mostly published by others, and have now built a database that includes 86 of the Saccharomyces genomes. And just to give you a scale, this is a subphylum, and so this covers about 500 million years of evolution and approximately more protein sequence diversity than you would get in the entire vertebrate subphylum. And so this is a tremendous amount of sequence divergence, a tremendous amount of metabolic diversity. And what you can see is that almost all of these clades have 0 or 1 representative that have been sequenced. And actually a lot of these are even unpublished, and we're collaborating in part with JGI to get those genomes out soon. But even once those are out, you still have a lot of clades with 0 sequences, and then most clades you have just a tiny narrow swath. And you can see where the focus has been is of course near Saccharomyces, where we again still have less than half of the species. And even here in the clade where Candida albicans is, we only have a tiny fraction of those highly relevant pathogens. And what's not on this are over 400 species that we don't even know where to put them, especially from the anamorphic genera. And so this is gonna, I think, be a really exciting time to start to answer questions like this. And I just want to highlight that it doesn't stop with aerobic fermentation, as cool and useful as that is. Some of these yeasts do potentially much more useful things, and just like our liver and adipocytes excel at accumulating fatty acids, the Uralia clade and Lipomyces, some of these species will accumulate over half of their dry weight as fatty acids or oils. And so these are potential target molecules for next generation biofuels. Many of the species in this clade actually ferment xylose, and so they've learned how to actually ferment and take this pentose phosphate pathway and make it into a champion catabolic pathway. So where does this project stand right now? Well, we have a website, which is nice. We have a picture of various yeast products where yeasts are involved, and this is in collaboration with Antonis Rokas, and Cleet Kurtzman is sort of the longtime granddaddy of yeast taxonomy, and he is at the USDA in Peoria, and so the strains are about 1/3 of the way being transferred up I-39 to UW-Madison. They actually use FedEx, but I assume that they go up I-39. So just to, again, just to give people a little bit of impressions that these are, a lot of these yeasts are probably not what you're thinking of. Here we're talking about, again, the Saccharomyces yeasts. There are a bunch of other independent times where fungi have lost multicellularity and have evolved the yeast lifestyle. We're just talking about the most speciose group that are related to Saccharomyces. But you can see, even within this group, we have a lot of really odd things. We have these yeasts that have these little hairs. This is the Trichomonas or Blastobotris clade. You have other ones like Lipomyces that look very oily. We don't think it's actually oil that's causing that. We think they're making extracellular polysaccharides, but all good microbiologists know you turn your plates upside down. If you do that with these guys, they just slime and ooze right off of the plate. And so there's a lot of really cool stuff going on. But most of my lab right now actually works on Saccharomyces. And of course, I think you probably don't, you've probably heard people from the Whitkop and Zhang lab probably tell you ad nauseum about the power of yeast genetics and how you can do anything. And it's true, you can do anything if you're willing to work at it. It's an amazing model system. You've got a small genome. You have complete environmental control. You have complete genetic control. And it's great. And indeed, the work that George told you about from my graduate career would not have been possible if it weren't for our friend, the super yeast. There, of course, are other benefits. You get to have the best smelling lab in the department, which doesn't hurt, although having flies nearby can cause problems, obviously, because the flies share your opinion. But as George alluded, my interests have shifted, although I think it's a medium-term detour, and that's because I think one of the most fundamental problems we have is actually what has been blocking Saccharomyces as use for a model for ecology and evolution. And I think it's actually the focus on this stuff right here. Not just because yeast researchers drink too much, but because it has caused us to focus not on the natural habitats and has created very limited and biased strain collections that have sort of misled us about what yeast biology is really all about. And so I think until we get a better strain set and better idea of what's out there in nature, then I think we can start to go into really going to town on this as an evolutionary model system. Did I stand too still? All right, lights out. Okay, so part of how we're doing this. So one of the things that's really exciting for me is that part of how we're going about improving our understanding of yeast biodiversity really involves undergraduate researchers, high schoolers, and high school teachers in a very real way, because it turned out this is a very labor-intensive process. Yeasts are, in most environments, very low, at very low abundance, and so we actually have to get them by enrichment. It turns out the very process of doing that sort of inherently teaches aseptic technique, and it also inherently goes from very tactile, concrete, to increasingly abstract, as you can see. And so so we take 18, 19-year-olds usually into the lab who've never been in a lab and have them dig in the dirt, and usually within a semester, they learn how to go through an enrichment protocol where they treat samples with lots and lots of sugar, so we give them about 8% glucose, limited ethanol, limited oxygen, which gives the yeasts an advantage over filamentous fungi, and we also treat them with a suite of antibiotics to kill bacteria. You do that a couple times, put them on plates, and you can see colonies. You introduce cell theory. And eventually, by the end of the semester, students have usually done PCR, extracted DNA, and identified by blast. And so we get a bunch of different things. We've isolated almost 100 different species, which is about 10% of known yeast biodiversity. And of course, we find new things. And when you find new things, you get to name them. We just published a paper with 8 new species. This is Blastobotrytis buckinghamii, which is named for our school's mascot. And I'm embarrassed to say he is actually from the UP. So he's technically a Wolverine, I suppose. But and you can see we named it for Bucky because this has a very aggressive hyphal stage. And so, again, this was just pulled out of Tequamanon State Falls, Tequamanon Falls State Park. And so, just to stress how little is known about yeast biodiversity, well, 8 new species is about 1/2 of 1% of all previously described species. And there are more species that are underway. Okay, so back to the main show. Here's so if we go back in time just a little bit, times were simpler about 15 years ago. We were worried about presidents and interns and what was going on, and we're less worried about ISIS and who was wearing dead animals on their head. In this era, unfortunately, the Saccharomyces genus is not such a great comparative model. And indeed, what was known is that there were Cerevisiae and its sister species Paradoxus. A popular model at the time actually was that Cerevisiae was simply a domesticated version of Paradoxus. That hypothesis has been thoroughly demolished by Justin Fay and Gianni Litti and Ed Lewis and many other researchers. But at the time, this was very popular by the hypothesis articulated by the Martinis. You can't make up these names. There was a husband-wife team called the Martinis from Italy. And then there was a mystery taxon that will come back to Saccharomyces bayanus. Again, not a whole lot. If you want to study sort of ancestral state reconstruction, any kind of inference, not a whole lot going on. With the adoption of molecular techniques, a lot of things got clarified. A lot of species turned out not to be species. And 10 years later, things got better in a lot of ways. And we're starting to have a plausible tree that you could imagine to start to do some types of comparative approaches on. There's still this bugaboo about what's going on with this bayanus taxon, and it turns out the solution to what's going on with the bayanus taxon is actually intimately tied up in various fermented products. And so, what it turns out is that a lot of what we were looking at here, what researchers were looking at, turned out to be a complicated web of interspecies hybrids that we're actually still trying to disentangle today, because there are a lot of things that have genes from different places. And in particular, you've got things that are giving rise to champagnes and ciders, but perhaps the biggest dog in the fight is actually lager beer, which for those of you that don't know, and many people have forgotten because of the rise of craft brewing and ales, 95% of beer in this country is still lager beer. Maybe not 95% of the good beer, but 95% of the beer is lager beer, and it's over a $250 billion industry in the US alone. And it turns out it is brewed with an interspecies hybrid, not with Saccharomyces cerevisiae. And of course, everybody expected, well, if we sample enough breweries in Europe, you'll find it. Well, no. So this is a global map of research and development dollars. This is, of course, mostly a map of spending on defense and biomedical. Very little of it is the most interesting and important kind of research on yeast biodiversity and taxonomy. But it turns out that if you look at where the major yeast collections are, I'm just going to argue that most of them are in the northern hemisphere. I think you can see that that's where most yeast researchers are. It's not necessarily surprising. And it turns out that the ancestor of lager brewing yeast that hybridized with Cerevisiae was found by our collaborator, Diego Lipkind, down in Patagonia, the very tip of Patagonia. And it turned out to be very prevalent there because it grows on these sugar-rich galls. These are Setaria herotii. This is a filamentous ascomycete, a Mycotina that actually taps into the phloem of trees, and these things become very sugar-rich. In fact, people will still serve them on salads as a little seasoner and sweetener. And in pre-Columbian times, the Aboriginal peoples would actually do an extraction for sugars and actually would ferment whatever happened to be there. And of course, now we know one of those things was Eubayanus that was probably doing that fermenting. I think now they just drink Bud Light. But anyway, so to cut a long story short, through genome sequencing and by comparing to a lager yeast genome sequence that had been published a couple years before, we concluded that the genome sequence of these South American isolates was 99.56% identical. And so because there was no non-hybrid version, we had to describe it, and I think this led to one of the most interesting anachronisms of fungal taxonomy, and that is Sequentia Genomo Totae Incibi Numero, and so that is a Latin description referencing GenBank as for the complete genome sequence. And this is, so we think this, the ironic thing is 2 years later, the rule that you had to describe things in Latin went away. You used to have to describe things in Latin so that your learned colleagues could understand it. Of course, now you're allowed to describe it in English. And I'm happy to report Fred Dietrich at Duke recently described the species in English, referencing the genome description as it. And I think those are the first two cases in fungal taxonomy, although I'm sure there are others in the pipeline. I know we have a couple papers we're getting ready to submit that actually use that as the legal description. But and I think that is, you know, how things will happen in the future. Okay, so our modern understanding of the Saccharomyces genus is that there are 7 naturally occurring species, and if we sort of look at the genetic sequence divergence, by the time you go out to the sister species, Paradoxus, you're at about human mouse level of protein sequence divergence, and by the time you get out to here, you're at about human bird level of sequence divergence. And then Uvarum and Eubayanus are the closest relatives. They have some biological reproductive isolation, but it's not absolute. And they're at about human macaque level of sequence divergence. Now it turns out that it's not just about Eubayanus and Cerevisiae in the fermented beverage industry. So by having these wild representatives, and then also by comparing them to genome sequences of other yeasts, we and other researchers have shown that many Belgian beers and cold fermented wines and ciders are actually hybrids of Cudrevzevii and Cerevisiae. Cudrevzevii, it turns out, is fairly prevalent in Europe, and so it's pretty easy to imagine how this wild species that has a nice cold tolerance ends up in hybridizing to get a cold fermentation beverage. Turns out that a lot of champagnes, through a story I don't have time to tell you about, a lot of champagnes turn out to mostly be Saccharomyces uvarum, another cold tolerant species, and these have picked up introgressions, interestingly, from up to 3 other species. And so one of the things we're seeing is that wild lineages don't have a lot of introgressions, and domesticated lineages often have introgressions, in some cases from many, many species. And of course, lager yeast itself is a two-way hybrid of Saccharomyces cerevisiae and Saccharomyces eubayanus. So how do these interspecies hybrids form? Well, they form, in order to think about how they form, we have to think a little bit about the yeast life cycle. And so yeast spend most of their time, 99.9% of their generations, and there are population genetic estimates that have made that for a few species. They spend most of their time as A-alpha diploids. And so I think a lot of molecular geneticists have sort of sold the haploid basis of yeast genetics, but that's actually something that we've done to laboratory strains so that we can work with them. In nature, they're spending most of their time as diploids, undergoing mitosis. And so the way you end up getting interspecies hybrids is that they're not nearly as discriminating as you and I, even though they're human chicken levels of difference by protein sequence. The mating pheromones are actually, in one case, identical, in another case, only differ by one amino acid. And so they can actually mate over quite large evolutionary distances if there's an A and an alpha that happen to come together. What doesn't work well is meiosis, but it doesn't matter because they can continue to undergo mitosis indefinitely as long as people happen to keep providing them food, which in the case of industrial hybrids is the next batch of beer or the next batch of wine. And so the fact that they can't undergo meiosis is not a major detriment to us to being able to brew things. So, so we can look at genome sequences of various lager brewing yeasts, and here I'm just showing two representatives, one from something called the Varberg lineage of lager yeast, and another one from something called the Saaz lineage. And so these are just named for the geography. These tend to be have originally been Czech associated, and these were originally Bavarian associated. And what you can see if you look at the plots, and these are just chromosome number variant plots, and this is something we routinely do now for all the Saccharomyces strains that we sequence, is we look at all 7 reference genomes and we look at the copy number so that we can tell whether they're clean or not. And SPIDER is just the clever acronym, or my student thinks it's clever, that Quinn came up with species plural SPP identification so that we can quickly tell who contributed. And so what you can see is that there are 2 quite different lineages of brewing yeast, and they have different properties, and brewers actually classically identified them correctly based on phenotype without genome sequence data. And so it's become somewhat controversial about whether these originated through multiple hybridization events or whether there might have been a single hybridization event. And sort of the best argument for the multiple origin, the hypothesis, has been the fact that there are distinct karyotypes, that you have here allotetraploids and here allotriploids. And it's a reasonably good argument. But Jürgen Wendland, and then actually another group, Daniela Del Neri's group, who actually took this side of the argument, discovered that there were actually a handful, in this case 3, breakpoints that are actually shared, and you can start to see where there are chromosomes missing in various places, and so you end up with a eubayanus chromosome fused to a Cerevisiae chromosome. And there are 3 that are actually shared between these 2 lineages of yeast, or the Varberg lineage and the Saaz lineage. And so depending on how, and so in this case when I say shared, I mean identical down to the base pair. Now of course if you're going to have a breakpoint you have to have a certain stretch of identity, and so saying down to the base pair really means, you know, down to the 100 base pairs or however long that stretch of identity is. But how improbable is it that you would get 3 pretty close ones? Well, I'm going to argue that maybe one of these doesn't count because this is the target of the HO locus, which is a known hotspot. This is actually deliberately cut by yeast in the mating cycle, and so you're actually cutting this every generation. But even so, you have 2. So what we wanted to ask is, well, is there evidence that maybe there were multiple parents? And so a student of mine, Emily Baker, came up with this test and reasoned that, well, if we look at the neutral site divergence, synonymous site divergence in Saccharomyces cerevisiae, and if we hypothesize that the hybridization event, that there was a single hybridization event, then that means it would have occurred before those 2 lineages started to diverge. And once those 2 genomes are in the same nucleus, they should start to accumulate changes at the same rate. Now, if instead what happened is that there are multiple hybridization events going on, you could actually have a situation where one of the genomes had begun to diverge earlier than the other one, and you might have a situation where if you look at the synonymous site divergence, that it's not going to line up. And so it turns out when you do that test, the answer is pretty clear. The two genomes don't line up. The Cerevisiae component of the genome is much more diverse, and it's not just a few loci, it's spread across the genome. I'm showing you the 10 gene windows plots of synonymous sites. You would get the same result, except actually with more statistical power if you look at individual genes. And so this is a clear signal that there were multiple Cerevisiae parents and multiple Cerevisiae parents. I almost stole the punchline from 5 minutes from now that there might be multiple Eubayanus parents too. But the only reason this test works is actually because it happens that the Eubayanus coalescence is more recent. And so that really gets to sort of the biogeographical problem of why did we discover this thing in Patagonia in the first place where it maybe shouldn't have been. So to summarize a lot of really hard work by a lot of groups, I'm just going to say that we have Northern Hemisphere isolates. This Fengyun Bei's lab in China has really been going to town sampling China for wild yeast and actually has across a broad swath of Western China and Tibet a number of isolates of Saccharomyces eubayanus. Our undergraduate-driven team has isolates from North Carolina, Wisconsin, and Washington State. And then we have a few that we haven't analyzed genetically yet. And then Christian Landry's lab also has a similar scale sampling effort in Canada, and he actually has collaborated with us on analyzing some of his strains. And so the score, if you're keeping score, is 14 North America, 10 Tibet, and still 0 Europe. Yeah. Absences it. It's just what we found. So we can't. So because we have to do this by enrichment, we can't definitively prove an absence of things. And in fact, Saccharomyces tend to be very poorly represented in environmental sequencing data, and so which I think is an interesting mystery of itself. But I will tell you that there are at least half a dozen different groups who have sampled in many, many different places in Europe, and so far everybody has struck out. Now, and some of those have consisted of dozens of samples that have been sort of well designed to pick reasonable places where you might find it. The one place you can succeed is you can find a bunch of hybrids that have eubayanus genes in the brewery, but that doesn't help with the problem at hand. Okay, so unfortunately, the Chinese strains are not available for general study, but we can look at a multi-locus data set that was published, and we can compare it to several of our strains, and we start to see some interesting things. One thing we see is that there are two major Patagonian clades, and they're both quite diverse, and we call them Patagonia B and Patagonia A. And then what we can see is that there's another clade here that includes the Chinese strains, and these cluster with the ancestor of lager brewing yeast, and then also with these North Carolina strains. So strains from the Northern Hemisphere, one of them actually falls within the Patagonia B clade pretty clearly. There's an exception that looks like a very recent admixture event, and that includes all of the Wisconsin strains and all of the Canada strains are nearly identical in sequence. And I'm going to show you evidence that they're actually the product of a single meiosis followed by haploselfing. And then finally what we have is this low diversity collection of strains that come from the whole Arctic region across North America, Asia, and then also Europe. The interesting thing, if you look at these different populations, Patagonia A, B, and so forth, and then also this distant subspecies, the only one that has anything really unusual going on is that the Tibetan lineage itself has a substantially negative Tajima's D, which could be an indication of a recent population expansion. You can also see it has very little diversity relative to either Patagonia A or Patagonia B. Okay, so when these strains were published, the sequence reads for one strain were deposited on GenBank, and so we were able to download those reads and actually build a whole genome phylogeny. So this is just a whole genome SNP phylogeny of a few of our strains and their strains. And so what we can see is that the Tibetan strain is actually the closest relative to the Saz and Froberg lineage. And you can see the Saz and Froberg lineages are almost identical. And of course, because you have whole genome data, you end up with 100% bootstrap support values because you have hundreds of thousands of SNPs, and you end up with just a sort of majority rule type of phenomenon. So one of the questions we wanted to ask is this true? Is this the whole answer? Is there a simple linear relationship like this where the Tibetan strain is closest and the North Carolina is a little bit further out? So we can look at sort of pairwise divergence plots. This is against the Saz genome, and what we can see is sort of like I already showed you, that the Froberg genome is very close to the Saz genome, although you can start to see there are a few places where they maybe do disagree. You can see Patagonia A is of course not a good match. This is the original where Eubanus was discovered. Patagonia B is a little less than 0.5% divergent, and it bounces around a bit. And then what we see is the Tibet and North Carolina strain are close to each other, but we can see the source of that signal. The majority vote clearly is that the Tibetan strain is closer, but we can start to see some parts of the genome where maybe there's a bit of a different signal. And so I'm going to introduce a type of plot that I think makes it a little clearer to look at, and I'm also going to go back to talk about those admixed strains as a way of introducing this type of plot. And so what we've done is we've just taken those pairwise divergent plots, and we now plot them against two different reference strains. And so in this case, we're plotting against a reference strain from A and a reference strain from B, and what you can see is we end up with what looks like a sort of a zigzagging mess where one goes up, the other goes down, and so on and so forth. And you can sort of track it, and you could say, well, this is that, and there's a crossover. But if you instead take those and do just like you would do with a microarray and sort of divide those two signals and then take the log2 ratio, that makes the 0 the part where it switches. And so now you get a really clean point where you can actually go and see the breakpoints and see where the ancestry flips from A to B, being more like A to being more like B. And when you go and you count the number of crossovers up, you get 45. Does anybody remember how big the yeast genome is in centimorgans? Trisha might remember. It's 4,800 centimorgans, so you expect exactly 48 crossovers. And so we're pretty close to what the expectation would be if there was an A parent and a B parent that had undergone meiosis and then haploself. The interesting thing is these 7 strains are nearly identical. You can count there's 10 to 20 differences between each strain. And so this is a very recent hybridization event between these two populations that ended up somehow in both Sheboygan, Wisconsin, and New Brunswick, Canada. And I've never been to New Brunswick, Canada, and the Landry lab did not have our strains before they discovered. So it's definitely not us. Okay, so now we can use these plots to perhaps ask more weighty questions rather than what's going on with this unusual ancestry. And so specifically, we want to ask if that general genome-wide signal that the Tibetan strain was closer is true across the genome. And it turns out that it's not quite true. And so if you look at, there are 20 different loci where you actually see the signals swap, where the North Carolina strain is actually a closer relative to the parent of Saz lager brewing yeast than the Tibetan strain. So again, the dominant signal is most parts of the genome are closer in the Tibetan strain, but the North Carolina strain has a few representatives. And we can permute the genome to sort of establish what is statistically significant, and that's where you end up with the red bar, and that's what tells you that's statistically significant. So the message is, and this maybe should not be that surprising, these are sexually reproducing outbred strains. The message is that there is probably no extant strain from the wild that is going to be the ancestor at all loci. We should expect, once we find the population from which alleles are being drawn, that there's going to be some variation at those loci. Okay, so what I think perhaps is most interesting about this is these parts where they disagree also means that if there were multiple parents going into Saaz and Froberg lager brewing yeasts, you might actually have a situation where different lager brewing strains actually drew different eubayanus alleles also. And so again, here is the genome-wide phylogeny. And if we make those same log2 plots, but now we color them in yellow and red for the different parents, we can see that, again, Saaz and Froberg were very closely related to each other, but we start to see a few cases where there's indications of different ancestry, and then we see one really striking case where there's an exact opposite. And we can go in and we can inspect this region manually and make sure there's no assembly artifacts, and we can even build conventional trees, and what we see is that you get really strong support for the Tibetan allele looking very similar to the Froberg allele, whereas the North Carolina allele looks very similar to the Saaz allele. And so we think that this is a strong indication that there is actually still some diversity from this low diversity Holarctic population that's actually present in modern lager brewing hybrids. Again, there's very little diversity there, but some of that wild diversity has actually made it through that hybridization barrier. Okay. So we can ask some more provocative questions and maybe come to less provocative conclusions. And so one thing we might want to ask is when the coalescence of this northern hemisphere population, as these Tibetan and North Carolina strains, is. And you could imagine some really trivial, silly explanations, perhaps. And indeed, the media was especially fascinated by this one in 2011. We probably encouraged it more than we should have. That perhaps a reason why people hadn't discovered it in Europe is, maybe trade was to blame. Maybe it's Magellan's fault. When the strains were discovered in China, the paper argues specifically that the Silk Road might have played a role. And so, well, what are you going to argue if you have strains from North America now? Well, it's got to be Columbus, right? So what I'm going to tell you is, if we make some reasonable assumptions and sort of look at how much sequence variation there is, and this is assuming what I think are pretty conservative assumptions. Actually, we're taking the maximum growth rate in 2% glucose, so we're giving them unlimited glucose, and we're growing them at 8 degrees Celsius, which is the average temperature in Patagonia, Tibet, Washington, Wisconsin. It's a bit warmer in North Carolina, but I still think it's pretty conservative. So I would argue the real dates are probably older because cells are not encountering 2% glucose all of the time, growing nonstop at their maximum rate all the time. And so if you do that, what you get is a coalescence that pretty decidedly, for this northern hemisphere population, pretty decidedly predates any kind of trade. So that's the downer. So what do I think is actually going on? And I'm going to say, I don't think we know for sure, but I think that the answer to how eubayanus is structured does not require, the population of eubayanus is structured does not require human activity. And instead, I think that there are geological forces at work. And the reason, in part, is that if we look at the sister species, Uvarum, we see exactly the same degrees of divergence in structure. We see two highly diverse Patagonian populations that have similar levels of diversity to these two populations, and we see that they're about 1% divergent from one another. Similarly, much like this West China outgroup population, we see an Australasia outgroup population. Interestingly, in a study I didn't have time to show you, the collaborative study with the Sampaio, Gonçalves, Lipkind, and Marullo labs, we showed that about half of these strains from this northern hemisphere, this Holarctic population, about half of these are wild isolates, and about half of them are domesticated champagne and cider isolates. You can see these have very low diversity, just like the Holarctic population. It's interesting that people have been able to isolate Uvarum going all the way back to the 19th century, so a lot of these are actually from very old collections, from wild collections. And for whatever reason, it's been just a lot harder to uncover wild isolates of eubayanus. But I would predict that we're starting to see some of the same patterns come up in terms of a low diversity Holarctic population. And in this case, the really cool thing is that the strains that have picked up alleles from other species, those are the human-associated champagne and cider strains, not the wild isolates. So I would argue that what we're starting to see is evidence that the eubayanus population structure is starting to look like the uvarum population structure, in that you have these two clades and then a low diversity Holarctic population. And so, since we've shown, I think, a reasonable inference that there was some variation in that Holarctic population, and some of that variation made it into the European isolates, I'm going to argue that we can probably indirectly infer that there probably is either is a European population, or there was in the recent past, that has some shared alleles with this population that we see in Tibet and North Carolina. And hopefully, someday somebody will discover it. Okay, so I've shown you some of the most interesting cases of, I think, of places where we're starting to understand where wild populations of Saccharomyces are, how they're structured. We've talked a little bit about how they evolved and how they differ in that there are interspecies hybrids, and that there's a lot of introgression between species. But I also I want to very briefly recap, and this summarizes a lot of published and unpublished work that we've actually now filled out the tree substantially for many of these species. And in most cases, we actually have considerably more genetic diversity than Saccharomyces cerevisiae. And so each of these distances is about 1% sequence divergent, and then these are 3 to 4% sequence divergent. And so we're starting to get a nice stepwise distance if you want to think about doing models of evolution of gene function or sequence function. You can start to think about doing that in Saccharomyces in a way that you really couldn't have even 5 years ago, because you have appropriate wild isolates that are coming from these different places. And really the only way it's been possible is people out collecting in the field and using molecular and genomic techniques to actually figure out what they have. So so how are we going to turn these into experimental models? Well, one option would be we could make auxotrophies for all of them, so that we can use all the standard tools of yeast genetics. And indeed, we sort of started doing that for a select group of them. And in fact, we maintain a set of auxotrophies for 5 of these species. And I think we've sent them to George and Tricia. And those are great resources. But at the rate at which we were discovering new isolates and others were discovering things, we sort of got worried that we really needed a more general solution so that we could work with anything. And so in order to address that, a postdoc in the lab, Bill Alexander, decided to develop a pan-fungal genome engineering device. And so what he wanted to develop it around was a marker that was both selectable and counter-selectable. And this sort of gets you the biggest bangs of yeast genetics. The most useful marker, the one you would make first, is Euro3, which has these features. And of course, double-strand break generators are very important to how DNA repair works. CRISPR-Cas9 is, of course, a very famous example of a double-strand break generator you can provide in trans. But what Bill did is actually make one in cis, so that it's a simple, easy-to-PCR device that contains a selectable and counter-selectable marker, and then a double-strand break generator that can cut itself and be induced. And so Bill developed the chemistry that allows you to use this novel selectable marker based on thymidine kinase from herpes simplex. And so you can make an anti-folate cocktail that allows you to select for the presence of the gene. And then if you don't have the gene, all of the Saccharomyces die. And then by giving it a toxic base analog, you can get exactly the opposite result. And so now we can kill everything that has it, and everything that doesn't have it lives. So what that allows you to do is to now go into the genome and put your herp cassette, and because it's a nice 3.2 kb piece, you can PCR across, and with 40 base pairs of homology, you can put it into a target locus. And so now you've replaced your gene of interest with a cassette And then because you have a double strand break, you can then get really efficient repair across that locus. When you do this, you then end up with a perfect, clean replacement at the chromosome target of interest, where you replaced whatever allele you started with whatever you wanted. And so by just doing a regular transformation reaction here, you can get a pretty good success rate. And here we can get about 100,000 independent transformants per reaction. So one thing we are starting to do with this is transforming in pools of alleles so that we can study the function across a phylogeny. And again, we're generating a strain that is otherwise isogenic, and we're only hitting the gene of interest. And this is just to show you a sort of early experiment that Drew Deering did, where he simply transformed in all the Saccharomyces alleles and randomly picked, and you can see that we get a pretty even representation. And Drew is now working on applying next generation sequencing to track these, as well as tracking individual mutations. Okay, with the last few minutes that I have, I want to talk a little bit about some of our biofuels research. And one of the things I want to highlight is that I've sort of shown lineages that we know are contributing to the beverage industry at least as hybrids. And a lot of that is sort of an accident of what happened to end up in Europe, and who happened to end up spreading their habits of beverage consumption around the world. But you can see that there are a lot of these Saccharomyces lineages, as far as we know, aren't contributing to anything useful in society. So can we put them to work? Well, so we're a part of a bioenergy center funded by the Department of Energy. And the main goal of the Great Lakes Bioenergy Research Center is to try to figure out more efficient ways to take the woody parts of plants, the plant cell wall, and turn them into ethanol from the undigestible parts, or advanced biofuels, whether it's isobutanol or perhaps even some of those fatty acids I talked about. So the problem with the process is you can grow lots of plants, of course. Plants are good at growing plants, or farmers are good at growing plants. And you can pre-treat them, and we can actually take apart the cell walls in increasingly novel and devious ways. But all of that ends up creating this toxic soup called a hydrolysate. And what's in that hydrolysate is a mixture of glucose, xylose, and lignin. So how does Saccharomyces cerevisiae react to these things? Well, pretty much like this. It loves glucose. That's what it's evolved to do, is ferment. It completely ignores xylose. Of course, that's something that can be engineered around. You can add enzymes in to get them to use xylose. But then you have this serious problem where these phenolic acid derivatives especially, or in the case of the basic pretreatment that GLBRC uses, they're actually amidated versions of these phenolics. These things are very toxic, and indeed, if you take hydrolysate and try to grow a laboratory strain of Cerevisiae, it just flatlines. And so, to summarize 5 years of work before I got to the GLBRC, they had created a hydrolysate-tolerant xylose fermenting strain of Saccharomyces cerevisiae, and that is our benchmark on 0. So what we did, so we're now comparing the growth of a bunch of strains to this benchmark of an engineered, evolved xylose fermenting strain. And what we can see is that Saccharomyces Makate, in particular, actually outperforms and grows faster in hydrolysate than the benchmark strain, by quite a bit more, a little more than 20%. The other strains, interestingly, not such good performers. The cool thing is, if we now take that already evolved industrial strain and hybridize it, many of these traits turn out to be dominant. So here's the Makate Cerevisiae hybrid. You now get the best of both worlds. You have that xylose fermentation cassette, and then you're preserving the hydrolysate tolerance. And indeed, we can start to see here the hydrolysate tolerance is being conferred by the Cerevisiae parent, so you get a big shift, and then this one ends up almost as good. So what we wondered is, can we now take these hybrids and sort of do exactly what the brewing industry has done for us? So if we take those allodiploid hybrids and now we sequence their genomes, we can see sort of the starting point is allodiploid, or appears to be allodiploid, And if we involve it for 50 generations, we start to see chromosome loss, aneuploidy. And you can see the loss of this chromosome ends up corresponding to the gain there. And so we're seeing some of the same patterns we're seeing in the brewing industry, but now these strains are adapting. And I can tell you that if you look, this strain has actually learned how to use more xylose than the engineered hybrid does. So it's actually adapted in hydrolysis to grow, to ferment xylose better. And this one actually looks even, this is a Cerevisiae hybrid. This one actually starts to look even, I think even more like some of the brewing hybrids in that you're getting some of the Cerevisiae genome is actually getting amped up rather than the Cerevisiae genome, and you see some evidence of translocations. The interesting thing is, if you recall, Cerevisiae is not one of the winners that we would have predicted. This one actually ends up doing as well or better than the parent after evolution, even though you wouldn't have necessarily predicted that by the screening. So I just want to show you sort of what I said here are some more of those spider plots. These are a bunch of different brewing strains, including some from a local brewery, just to show you that, these are the two evolved biofuel strains, and you see a lot of the same patterns in terms of aneuploidy and likely translocation involved. So the lesson I want to leave you with is that Saccharomyces cerevisiae, of course, has been a great model system, and I think that we have started to go a long way to really flesh out the rest of the genus in the way that it can now become a model, just not for mechanistic studies, but where we can actually ask questions about the biology and ecology and how these species differ from one another, and in a system where we can rigorously get to the molecular mechanism. And so, in particular, I think what's been important is going out and actually improving the collection. So we now have dozens to hundreds of each species in the lab from the wild, and so that makes a lot of things possible. And so the other thing to highlight, I think, is that we're starting to have come online general tools that allow us to do a lot of the same tricks that you would do in Saccharomyces cerevisiae with similar types of powers. And so hopefully it isn't just model a super SC, but super genus. And ultimately, I think being able to do these types of tricks even in a more general way beyond Saccharomyces. And finally, I've generally highlighted people on slides as I did the work, but I want to mention Bill Alexander has done a lot of the synthetic biology and tools development. David Paris and Quinn Langdon have looked at Saccharomyces eubayanus population diversity and population genomics. Emily Baker did the Saccharomyces eubayanus sequence analysis, and then of course undergraduates in particular have been invaluable to expanding our strain collection. And I'll take any questions people have. Thank you. Tricia.
In mating and mate recognition and many animal species are you multiple? Is there any models why the pheromones can use that?
Why are they so? Yeah, because so one reason might be that the mating pheromone itself is not that big. But that in and of itself shouldn't say why of the 13 amino acids one of those couldn't change. So I know Duncan Grieg recently published a study where he took a pheromone from a more distant yeast and sort of tried to model how many changes it would take to get it back to allow it to mate. And I think the answer was relatively few. But yeah, I don't know. Do you have a thought about why it's so conserved? And because you would think certainly if you don't want to mate, if mating ends up being largely a dead end, it would be. So I think probably they're not coming statistically into contact with other members of the species that much, and so there may just not be enough in order to get that sort of reinforcement going. So one thing I didn't talk about is in Patagonia we have enough samples from enough ecological sites. We have over 200 eubayanus and over 200 uvarum, and it turns out that while they do exist in sympatry, there's a statistical association that eubayanus tends to be found on one type of nothophagous tree and uvarum tends to be found on another. And so even though you can find them in both places, it's not an absolute association. there's probably some, you know, some micro allopatry.
And how does it differ from the hybrids compared to the present technology?
Well, so it depends on the environment. So, for example, in the case of Eubanus and Cerevisiae, it's been shown actually by a number of researchers that the hybrid does indeed end up having a temperature preference to the colder temperature, and so Eubanus is conferring cold tolerance, and then it's also and we've seen it as have several other groups. The Eubanus isolates don't seem to be able to use maltotriose, which is so maltose is two glucoses hooked by an alpha linkage, and then if you have three, it's maltotriose, so it's just an incomplete breakdown during the malting process. And it turns out that is the second most common sugar in wort, and so Eubanus is not able to use maltotriose, the second most common sugar, but Cerevisiae is. When you hybridize them, the Cerevisiae trait is dominant. We have some candidate genes we're actually testing to sort of to see what the genetic basis is for that. Any other questions? George?
So you talk about lots of hybrids, and you say that they are fine in mitosis, but they have trouble in meiosis.
Mm-hmm.
So do you think they can live on forever? I suppose that
Forever.
when they're under stress, they will have trouble with mitosis.
Yeah, so I think so if you can't do meiosis, then you become susceptible to Muller's ratchet, right? You're no longer able to purge deleterious mutations, and you're also potentially denying yourself any adaptive mutations that others have acquired. And so I agree, there probably is a limit. Now, if they lose enough chromosomes, they might start to come back to a more euploid state, and indeed that's thought to be a big part of sort